thermostable enzyme system, thermozymes, and application involves biofuel production, food and baking, animal feed and enhancement, and waste treatment. So this is already got patent actually. So there are other research R&D is going on with respect to marine fungi. For example, Antarctic marine yeasts are used for cold active lipase as a proteases. Aspergillus flavus is tested for thermostable amylase. One more fungus is used for salt tolerant lacase. Penicillium citrinum from the marine source is tested for hydrocarbon degrading enzymes. Clematis versicolors, these is probably not a very strictly marine fungus, probably it's a marine derived fungus. It is used for lacase, again, all those things. There are some problems, if you want to collect samples from deep sea environment, it's not easy. You need a ship, and ship will cost you 15 lakh rupees per day. And not only 15 lakhs, you need to have expertise to collect the samples and process it. You get the sample from 4 or 5 kilometers below the sea surface, below the sea surface. Once the sample comes to the normal laboratory condition, the conditions are very different. If you're talking about 5 kilometers below the sea surface, that means 500 atmospheric pressure. We are at one atmospheric pressure, right? So much of pressure, the temperature is 4 degrees Celsius there, and you may not get all the time get the food. There is a food and famine situation. Sometimes there is no food. Sometimes lots of food coming from the top. So getting sustainable supply for industrial application is a problem actually. And even if you develop something, you need to actually compete with the molecules from the terrestrial origin on cost and efficiency. There are regulatory, you cannot just use marine fungi just like that. There are regulatory requirements too. And how we can overcome, we can use genomic mining and engineering. So if you use when you use genomic mining and engineering, that can uncover and activate silent gene clusters in existing fungal strains. So that will help you in accessing the chemistry part of it. You can use synthetic biology. So because culturing marine fungi, especially from deep sea environment, is very difficult. So you can bypass the culturing part. You can use the synthetic biology. You can transfer some of the genetic material in E. coli or Pichia ion system for reliable production of some target molecules. Advanced bioprocessing, like if you can use bioreactors. You use culturable fungi plus bioreactors, so that can mimic the natural conditions. And it's okay. How much more time you have, ma'am? I have, ma'am. Yes. 12 minutes only. 12 minutes only. Okay. Okay. I'll take 3 more minutes. Yeah, thank you. So this is about the marine fungi. Like we thought about it. We discussed among the people who are working in mycology. So what are the things we need to focus on? First one is many of the laboratories are not equipped for deep sea culturing, deep sea fungi. So especially high pressure culturing. So we think, we believe that these facilities should be upgraded, and there is a need. It would be great if you could create a national repository for marine fungi that could have a database for species, genetic data, and metabolites. It's good to have more funding. I mean, you can always try to get more funding, but more funding cannot be say, I want to study marine fungi, their taxonomy. Nobody will give money for taxonomy. You need to say, I am going to collaborate with chemistry people, biotechnologists, then probably chances are getting. You need a consortium. It cannot be मैं अपने lab में काम कर रहा हूँ, मुझे marine fungi पे काम करना है, मुझे पैसे दे दो. Nobody will give money, especially if you're not from the coastal areas. I have friends from Bhopal and all. They want to work on marine fungi. That's not easy. Your lab is very away from the marine setup. And the collaborative research, as I told you, we need to promote interdisciplinary and international partnership, capacity building. We need to train early career scientists in culturing, not only culturing, bioinformatics, NGS analysis. It is better to get industrial partners. Then impress upon the fact that these fungi could be a great source of novel molecules if you can consistently work on it for next 10 years. There are already some molecules in the market. Not really market, one in the market from the marine source, many in the clinical and preclinical stages. These are like, there are some bright spots in this discovery path. We could discover we can explore new habitats like mangroves, coral reefs, deep sea, and we can focus on culturing methods. Of course, there's a limit to which, these fungi are very stubborn. You cannot make them to grow in the cultured media unless and until they want to grow. So they have been living here in a condition. The temperature is 4 degrees Celsius plus pressure is around 500 atmosphere. They are very harsh condition there. And you say get them into the lab condition in your labs. They may not like to grow there. So, but you can mimic the natural condition using bioreactors. And the environmental initiative. One of the things I have been thinking is integrate mycology, marine mycology, into conservation, environmental conservation and climate change. If you use climate change word, there's a better chance of getting the attention of the funding agency. So by conclusion, I think I am going to touch 15 minutes now. Most of the marine fungi are still undiscovered. At least you can describe 3,000 new species of marine fungi. And what is the fun? They could be source of rare compounds that could be used in medicine, biotechnology, and maybe chemical engineering, actually. But growing them at a large scale is still a challenge because they don't want to grow in culture. So we can use bioreactors and genome mining and other stuff. And India is lagging with when you compare with China. India used to be very good in marine mycology. We had a great team in CAS botany and some of the other laboratories like Goa, Goa University and other. Now, people who have got trained in marine mycology got diverted into other fields. They are doing immunology, they are doing bacteriology. There are very few marine mycologists in India. So I hope we could get the attention of some youngsters and into marine mycology. And hopefully we'll have a better activities in coming years. I would like to thank the organizers. Thank you very much, Vinith. Thank you very much, and Shilpa Madam and other people. My organization, CSI NIO, and my collaborator, Dr. Rohit Sharma, and Professor Sridhar, and other people in.
Okay, sir, thank you so much for your nice presentation. Actually, this is not my field, but I am a little curious about that. You told that it is very difficult to get a fungi from 4-5 kilometers in from the seawater. So I am having a doubt that what kind of technology you are using through which you can get one special kind of fungi from 4-5 kilometers. Like suppose we are or your team will be doing real life.
No, sir. We cannot go there below 50 meters of this ocean. What we use, we take our ship, go to the open ocean. There only we can reach the depth of 5,000 meters. So then we have a pulley and they send a grab. So that the grabber will get the sediment.
Some raw materials will be collected.
Yeah, sediment, and in addition to that, we have water collectors also. So to go to that area, it will take 3 hours. ऐसा पानी जैसे कुएं के अंदर ऐसा नहीं होगा, स्लोली चलना पड़ेगा और स्लोली ऊपर निकलना पड़ेगा. तो ऊपर नीचे आने तक टाइम तो 4 घंटा लग जाता है. उसके देखना सैंपल का सिचुएशन क्या हो वो भी देखना पड़ता है. So it is quite challenging, but we have something called pressure vessel. It can maintain pressure up to कितना 400 atmosphere चाहिए वो maintain कर सकता है. और टेम्परेचर भी probably उतना ही हो सकता है एक्चुअली. बट तो ये सब इंडस्ट्रियल स्केल पे कैसे चलेगा? दैट इज ऑल्सो इम्पोर्टेंट. इसके लिए कल्चरिंग को छोड़ के, कल्चरिंग इज गुड, उसमें यू डोंट हैव टू ब्रेक योर हेड ऑन कल्चरिंग, गो फॉर अनकल्चरेबल पाथ. जैसे है ना दैट you get the genetic material, put the gene in the E. coli or Pichia and produce the molecules required. I think we need to unlearn many things. One of the things we need to unlearn is we should not be focusing too much on culturing with respect to deep sea fungi.
Thank you, sir. Is there any question from research scholar or any colleague or scientist or level here?
Yes.
Thank you, sir. Very nice. When we collect the samples, What you exactly collect?
We collect, in our deep sea research, we collect sediments and also water. And sometimes organisms living there and the associated microbes.
Sanjay, you are bringing it in water and they are also elements.
Yes.
And do you have also idea about if Sanjay also associated with some seeds?
वहां तो सीवीड्स प्रोबब्ली नहीं मिलेगा सर 5 किलोमीटर बिलो में तो सनलाइट ही नहीं आता तो सीवीड का मुझे नहीं लगता
Seed do not have associated.
नहीं उसी डीप सी में नहीं मिलेगा बट नॉर्मल इसमें मिलेगा एज एंडोफाइट्स तो बहुत मिलेंगे आपको एज एंडोफाइट एज ए पैथोजन भी मिल जाएगा सीवीड तो मोस्टली कोस्टल एरिया में दिखता है सर डीप सी मतलब मैं बोल रहा हूँ एटलीस्ट 1000 मीटर के नीचे ही जा रहा है वहां तो सीवीड्स मिलना मुश्किल है बट जो भी है वो जो जहां प्लांट है वहां एंडोफिटिक फंजाई एक्सपेक्ट कर सकते हैं विनीत से बात कर लीजिए आप ही इज एन एक्सपर्ट ऑफ एंडोफाइट्स
Sir, I'm having a simple basic doubt. Actually, it's very challenging to have sample collection, as you said, and we have to overcome. We require a lot of techniques to overcome such kind of challenges. अगर accidentally अगर हमारा कोई फंगस हमने कलेक्ट करके लाया वहाँ सैंपल्स और लैब में वो ग्रो भी हो गया, तो आफ्टर कल्टीवेशन हाउ वी कैन मेंटेन इट फॉर लॉन्गर टाइम? मतलब अगर हम नॉर्मल फंगस हम काम करते हैं तो नॉर्मली वी इंक्यूबेट एट 27 डिग्री सेंटीग्रेड एंड वी कैन सबकल्चर इट फॉर लॉन्ग टाइम. सो अगर हमको इसको मेंटेन करना है मराइन फंजाई तो इज देयर एनी स्पेसिफिक कंडीशन और टेक्नोलॉजी और मेथोडोलॉजी अप्लाई टू गो फॉर लॉन्ग टाइम ऑफ प्रिजर्वेशन?
There are some fungi which can be cultivated fairly easily because they are not very stubborn. They are not very obligate kind of thing. They are. But there are some. These are called facultative. But there are some fungi which refuse to grow unless and until you give them the temperature of 4 degrees Celsius and the atmospheric pressure of वहाँ कितना था फॉर एग्जांपल 500. वो इसके लिए एक इक्विपमेंट है उसका नाम है प्रेशर वेसल. ये बहुत पुराना इक्विपमेंट है वो लैब में हम कर सकते हैं बट कितना साल मेंटेन कर सकते हैं दैट इज बताना मुश्किल है बट बेटर वुड बी एक्सट्रैक्ट द डीएनए मेंटेन ए डीएनए बैंक maintain a DNA bank. Any unculturable fungi, maintain a DNA bank. Unculturable, difficult to culture. Yeah, उस टाइप का fungi को. Yeah.
Thank you, sir.
Okay, thank you, sir. Thank you so much.
Thank you.
Thank you so much. Can we have a huge round of applause for Science. To felicitate our speaker, Dr. Vinay Amudaj Kumar, with memento kit and certificate as a token of appreciation. Can you clap for him once?
Thank you. Thank you, ma'am. Thank you. पैक करके देंगे क्या? हो गया चाय पीना मुझे कैंटीन है क्या?
To move forward, now I would like to invite Dr. Vinay Sham sir, Dean RME, to welcome our esteemed speaker, Dr. Gautam Anand sir, to welcome him with a bouquet. Now I hand over the mic to Ayush.
अरे यार मेरे सूटकेस में जगह नहीं है इसके बाद मुझे बैंगलोर जाना है
Move to our next speaker,
बैंगलोर से पौंडीचेरी
Dr.
में
Anand sir,
होटल
who
जाना
is currently working as an assistant professor at the Department of Biotechnology
Sandeep University, Nashik. Dr. Anand obtained his
Wonderful
master's degree
reason
in
about the marine
downstream
fungi.
biotechnology from Green Mineral Biotech, Gorakhpur University. He has been awarded with the Jawaharlal Nehru Memorial Scholarship for doctoral studies. He has 8 years of post-PhD experience in overseas labs. He has also worked on various enzymes such as counterfectinases, particularly
चेकआउट किया है
polycalenterases and
तो एक ही दिन का था बुकिंग
his current research is
आपके शाम तक का तो चेकआउट करने का टेंशन नहीं है।
sensitive fungi and behind fungal genetic mechanisms
मैं जो बोले
plant fungus interactions. He has published more than 30 research papers and articles in journals of international repute, 19 book chapters and edited one book. So I would like to invite Dr. Gautam Anand sir on stage to share his valuable thoughts with us.
ऊपर में लगाओ ना अगर नीचे लगा दो
At the same time, an important announcement is there that, as we know, we are that race is always against time. So, sorry to say that you all note speakers have to finish their presentation within 20 minutes. And only one questions will be allowed to ask because time is always constrained. So, thank you so much.
A very good afternoon to all of you. After a nice talk by Shinaz sir on marine fungus, uh, we are moving on to the phytopathogenic fungi. Today I'll be presenting one of my research topics about understanding the response of fungal phytopathogen to cytokinin. The model fungus in our for our research was the Botrytis cinerea, which is a very devastating plant pathogen, having more than 1,000 hosts and following a necrotrophic lifestyle. It not only infects leaves, but it's an important post-harvest pathogen as well. It can infect. Recently, it was shown in China that they can infect even medicinal plants. The molecule of our research was cytokinin. It's a very well-known plant hormone having a role in plant development, senescence, nutrient acquisition, and recently our group suggested that they can induce defense response in plant against necrotrophic fungal pathogen by inducing plant defense pathways. So, just being curious that inducing the defense response is not only the pathway how it is inhibiting fungal pathogen. What we did was it was just a serendipity. We tried to look into the direct inhibition as well, like if the cytokinin could directly inhibit the fungal pathogen as well, apart from inducing the plant defense pathway. And to our surprise, we found that cytokinin, the plant hormone cytokinin, could directly inhibit fungal pathogen as well, apart from inducing defense response in the plant. And it was not only effective against Botrytis cinerea, which is a necrotrophic fungal pathogen, but it was effective against a wide range of pathogens following different lifestyle like Fusarium oxysporum, which is a well-known soil-bound fungal pathogen, as Rholsii, which is a necrotrophic soil-bound pathogen, and all were significantly inhibited by cytokinin, the plant hormone. The molecule, the cytokinin which we used in our research was 6-back, which is a synthetic cytokinin and a stable one. But we found that even the natural cytokinin like transietin, kinetin, it was also significantly inhibiting the growth of fungal pathogen. This slide shows the physiological level of cytokinin in the plant leaf. And if you look at the overall cytokinin content in the leaf, in the normal leaf, in tomato, it comes around 400 nanomolar. And in our research, we found that after the infection, 48 hours later, after the infection, the cytokinin content increases in the plant leaf. So, and for our research, we were testing at 100 micromolar. But what we found that even up to a level of 100 nanomolar, it was significant of cytokinin. It could be inhibiting the fungal pathogen. We looked at different growth parameters like Botrytis cinerea, sporulation, spore germination, germ tube length, and we found that all was significantly inhibited by the fungal pathogen, by the plant hormone cytokinin. You can see even at like 100 nanomolar also there was significant reduction on the growth parameters of the fungal pathogen Botrytis. To get a mechanistic insight, like what is happening at the molecular level when you are supplying cytokinin to Botrytis. So what we did, we went for RNA-seq analysis. We did transcriptomic data, and you can see a lot of cell cycle pathways were downregulated. The fungal cell cycle pathway. DNA replication was downregulated. Endocytosis was significantly downregulated. And we found a lot of virulence genes were also downregulated upon treatment of the fungus with the cytokinin. The virulence genes is not, they are not very well annotated in fungus, so we developed our own list of virulence genes, and we found that a lot of them were significantly downregulated. Since our transcriptomic data suggested that there was inhibition of the cell cycle endocytosis, to confirm it in the lab, what we did was we stained our fungus with calcofluor white stain and checked the cell area, the germ tube length, and we found that it was significantly reduced. Also, we did Hers staining to check the to check the nucleic acid content of the fungus, because cell cycle pathways were downregulated. So it should be reflected in the nucleic acid content also, and we found that nucleic acid content was significantly downregulated in our cell biology assays. A lot of cytoskeleton genes were downregulated. The blue color shows downregulation of the gene, and it was further confirmed by qRT-PCR, and we found that tubulin, actin gene, they are all significantly downregulated. Since our transcriptomic data suggested that cytoskeleton genes are downregulated in the RNA-seq analysis, what we did was we developed botrytis transgenic line expressing life-actin GFP. Life-actin is found at the tip of the growing hyphae, which is a hallmark of fungal growth. So what we did, we tagged the life-actin with GFP, and we used our transgenic botrytis line with the cytokinin. You can see in mock, the life-actin is localized at the tip of the growing hyphae. But when we treat with the cytokinin, the life-actin loses its tip-specific localization, rather it is diffused throughout the cell. So this could be one reason why when you are treating the fungal with cytokinin, there is a reduced growth of the fungus. Endocytosis pathways, their genes were downregulated. So to confirm it, we did FM464 staining of the fungal hyphae, which is a well-known endocytic tracker dye, and we tried to measure the size of the endosome and also the density, and we found that both of them were significantly reduced. But if you look at the literature survey, there are a lot of fungi, even pathogens, which are itself secreting cytokinin. So inhibition is not the only effect. There must be some role of cytokinin in fungal biology also. And if you do a phylogenetic analysis of the cytokinin biosynthesis gene in fungi, you'll see that there is a lifestyle-based clustering of the cytokinin biosynthesis gene. So there must be some role of this cytokinin even in the fungal life cycle itself. To understand it better, what we did was we went back to our transcriptome data, and what we found that a lot of energy metabolic pathways were upregulated. Glycolysis pathway was upregulated, starch metabolism was upregulated, oxidative phosphorylation pathway was upregulated. To confirm it, what we did, we prepared 4 different types of media, synthetic media. In one of it, where glucose concentration was 1/4, in one where the nutrient concentration was 1/4, and in one media, both glucose and nutrient concentration was reduced. To our surprise, when the glucose concentration is reduced to 1/4, the same cytokine which was inhibitory to the fungus, it was promoting the fungal biomass. Remember our life-actin botrytis line. What we did was we treated it life-actin GFP in full and 1/4 media, and then supplied cytokinin. you can see in full media that fluorescence at the hyphal tip is significantly low. But in 1/4 media, it's barely significant. There is some reduction, but it's barely significant. Since the glycolysis pathways were upregulated, to confirm it, what we did, we tried to see if it can rescue glycolysis inhibition cytokine. So we use 2DG, which is a well-known glycolysis inhibitor, and we treated our fungus with 2DG and cytokine. What happened when the glucose availability to the fungus is reduced? It was rescuing the glycolysis, which is suggestive that there is some role of cytokine in uplifting the glycolytic pathway when there is nutrient starvation condition. Even the glucose uptake from the media, from the 1/4 media, it was significantly increased. Any effect on the metabolic pathway should be reflected in the redox status of the fungus. So to confirm this, what we did, we prepared two botrytis transgenic line, redox line, one for measuring the mitochondrial redox and one for measuring the cytosolic redox. What we found that in 1/4 media, when the glucose concentration is reduced to 1/4, the mitochondria was oxidized and the cytoplasm was reduced, which suggests that there is an upregulation of the metabolic pathway. If to check whether in planta cytokine concentration could also affect the metabolic state of the fungus, what we used our transgenic botrytis line for measuring the fungal redox and treated with and we infected IPT transgenic tomato lines, which has increased cytokinin content in their leaf. When we infected the leaves of IPT line, IPT transgenic tomato line, with our botrytis, with our redox transgenic botrytis line, what we see that the mitochondria after 48 hours of the fungus was oxidized and cytoplasm was reduced. Where in the plant where the cytokinin concentration is high in the leaf, suggesting that even in planta cytokinin concentration could alter the redox state of the fungus. One could say that the effect of cytokinin on the fungus is due to the reduced uptake of the cytokinin in the fungal hyphae. So to null out this hypothesis, what we did, we used fluorocontact cytokinin and checked the penetration of that cytokinin in our fungal hyphae. It was found that there was no significant reduction in the cytokinin penetration. So whatever effect is, it's not due to the less cytokinin that is going inside the cell. Points are not fine. Okay. Okay, these are the concluding ideas. It's not appearing on the screen. So throughout the research, what we found that cytokinin effect on the fungus, it depends upon the nutrient availability to the fungus. It's not that it will always be inhibitory. It will depend on how much nutrient is available to the fungus. So there is a dual role in the life of fungus of the cytokinin. It could be inhibitory when there is high nutrient condition, but at the same time, if the concentration of the nutrient to the fungus is reduced, the same cytokinin could promote the fungal biomass. My sincere thanks to my research team, Professor Maya Bar, who was the PI, our mentor, my colleagues, Dr. Rupali Gupta, Mirav Liebman Marcus, our PhD student. A sincere thanks to our collaborators in Israel, in Czech Republic, in Chile, And last but not the least, a sincere thanks to the funding agencies without which this work was not possible.
Thank you, Dr. Anand, for this nice talk. There is any question from research scholar or any who are doing in PhD in this field? Please, one question is allowed only. There is.
Thank you, Dr. Gautam, for your nice talk. And I see that you have very good publications. My question is, can we use cytokine as to as like you use pesticide? So can we use cytokine as an alternate of pesticide? Is it possible, or fungicide?
It is possible because it not only inhibits the fungus directly, it primes defense signaling also. Like if you treat the plant with cytokinin, what you see, like there is an induction of the jasmonate or salicylate defense signaling pathway. So it could be used, but it should be cheap, cost effective. Because the synthesis, like in our one of our experiment, we used fluorochrome tagged cytokinin. It was very hard to get. And even this 6-bag, the synthesis synthetic cytokinin, it's quite costly. So we can develop something like this, which can induce defense pathway as well and can inhibit the pathogen directly.
Thank you.
Okay, thank you, sir. Thank you so much.
We sincerely thank you for taking time to share your thoughtful and valuable perspective with us. And now, may I request Mr. Dinesh Krishnani, sir, from Associating Faculty of Design, to felicitate our speaker, Dr. Gautam, sir, with a memento kit and certificate as a token of appreciation. Dinesh, sir, please stay you have. So, welcome our esteemed speaker, Dr. Shubha Mishra, ma'am. Please put your hands together to welcome our speaker, Dr. Shubha Mishra, ma'am, with a bouquet as a token of gratitude. And now I hand over the mic to Vedita. Next, we have Dr. Shubha Mishra, ma'am, who is serving as an assistant professor in the Department of Physics at Government Naveen College, Ludhiana, Rajasthan. She possesses more than 18 years of teaching experience in higher education. She is actively engaged in teaching, research, innovation, and academic development initiatives aligned with NEP 2020. She did PhD in physics from National Institute of Technology in IT. She has also worked on 25 research papers published in reputed Scopus, national and international journals. She has authored 6 books and published 5 book chapters. She has also participated in 35 plus national and international conferences, seminars, and workshops. So I request Dr. Shubha Mishra ma'am to come on stage to share her valuable thoughts with us.
Thank you so much. Good afternoon to everyone. Today my topic is patent awareness and intellectual property rights for sustainable development. Myself, Shubhra, and at present I am in Government Naveen College, Budhari, Raipur. These are the contents. Today I will cover it. What is innovation? What's the meaning of innovation? What is intellectual? What is intellectual properties? What is intellectual property rights? And types of intellectual property rights. Pillar of intellectual property rights are patent, utility patent, design patent, copyright, trademark. Then multiple IPRs in a single product, validity of IPR, offices in India in IPR, and our contribution in patent. What do you understand by innovation? Innovation का मतलब आप क्या समझते हैं? यहाँ कुछ स्टूडेंट्स बैठे हैं, मैं उनसे जानना चाहूंगी कि इनोवेशन का मतलब आप क्या समझते हैं? सिर्फ हम एक तरफ से बोल रहे हैं, आप सुन रहे हो, ये नहीं, आप बैठे हैं स्टूडेंट्स, तो आप थोड़ा सा इंटरेक्ट कीजिए। इनोवेशन का मतलब आप क्या समझते हैं? आपके सामने स्लाइड में लिखा हुआ है, आप कोई भी एक स्टूडेंट बोलना चाहे तो फिर ये वही बात हो गई कि हम यहाँ बोल, कोई भी एक स्टूडेंट, इनोवेशन का मतलब क्या है? यस, गुड। यदि हमारे माइंड में कोई भी न्यू आइडिया आता है तो हम वहां तक ही उसको इनोवेशन नहीं बोल पाएंगे। जब तक आप उसको एम्पलीफाई नहीं करोगे, आप उसको प्रैक्टिकली यूज नहीं कर पाओगे, आप उसको कमर्शियली यूज नहीं कर पाओगे, तब तक वो इनोवेशन नहीं कहला पाएगा। यहाँ पे लिखा हुआ है, आप देख रहे हो, क्रिएटिंग समथिंग न्यू आइडियाज इनटू प्रैक्टिकल सॉल्यूशन और इम्प्रूविंग व्हाट एग्जिस्ट इन मोर इफेक्टिव वे। इनोवेशन की जब हम बात कर रहे हैं, खासकर इंडिया में, हम बोल रहे हैं विकसित भारत बनाएंगे हम भारत को 2047 तक, जिसका एक पर्टिकुलर स्लोगन है क्रिएटिव इंडिया, इनोवेटिव इंडिया। हम बहुत दूर नहीं जाते, हम बात करते हैं अपनी ही कंट्री की। एक सिंपल सा एग्जांपल लिखा हुआ है, ऑनलाइन टैक्सी, उबर, ओला, आपने सुना है? ये सारा इनोवेशंस की बात हो रही है, मतलब ये सारा कुछ एप्लीकेबल है। इनोवेशंस के कुछ एग्जांपल्स आपके सामने हैं, जिसमें आप देख रहे हो कि एग्रीकल्चर फील्ड में आजकल हम ड्रोन के थ्रू दवाइयां डाल रहे हैं, हम ड्रिप के थ्रू पानी डाल रहे हैं। स्मार्टफोन से लेटेस्ट अभी आप लोग क्या चीज यूज कर रहे हो ह्यूमन हेल्थ को लेके? यदि हम इनोवेशन की बात कर रहे हैं, जब तक आपके माइंड में जो लेटेस्ट चीज चल रही हैं आप उनके बारे में नहीं सोच पाओगे, तो आप इनोवेशन करेंगे कैसे? ये कॉमन टॉपिक है जो आप लोगों के लिए है, खासकर स्टूडेंट्स लोगों के लिए है, कि आप क्या सोचते हो। हम हैकेथॉन कर रहे हैं, हम नए आइडियाज की बात कर रहे हैं। आप एक लाइन मुझे बताइए कि डिजिटल एजुकेशंस में लेटेस्ट टेक्नोलॉजी में क्या चल रहा है? आर्टिफिशियल इंटेलिजेंस और मशीन लर्निंग के थ्रू आप क्या चीज कर रहे हो अपने डेली लाइफ में? ये सारी चीजें आप लोगों के लिए सोचने के लिए है। तो इनोवेशंस का फॉर्मूला, न्यू आइडिया, क्रिएटिविटी, देन प्रैक्टिकल यूज की बात कर रहे हैं। एक छोटा सा एग्जांपल आपको यहाँ पे दिख रहा है। सपोज आप किसी फार्म पे एक इंटीग्रेटेड सोलर कोल्ड स्टोरेज बनाते हैं, जहां पे आपको सोलार पैनल की जरूरत पड़ती है। सोलार पैनल के साथ आप देख रहे हैं यहाँ पे आपको टेम्परेचर कंट्रोल करने वाला एक सेंसर लगा हुआ है। वो जो भी आप वेजिटेबल फ्रूट्स वहां रखेंगे वो उसको कंट्रोल करेगा। बैटरी का एक अपना इंपॉर्टेंस होगा। ये सारी चीजों के थ्रू आप अपने इनोवेशंस को सोच सकते हैं। आप मेरे सामने जो बैठे हैं सब एकेडमिक से जुड़े हुए हैं। हम रिसर्च करते हैं, हम पेपर पब्लिश करते हैं, हम प्रोजेक्ट बनाते हैं, हम प्रोजेक्ट गाइड करते हैं, हम मॉडल बनाते हैं, बट क्या इस मॉडल को आप लीगली अपने नाम पे कर पाते हैं? क्या उस मॉडल के मैकेनिज्म को आप समझ के आप अपने नाम पे कैसे लेके आएंगे? ये सारी बातें हम करते हैं पेटेंट को लेके, जहां पे मैंने बेसिक डिफरेंस के थ्रू बताया है आपको। इंटेलेक्चुअल इंटेलेक्चुअल मतलब माइंड से जुड़ी बातें, माइंड की बात बोल रहे हैं हम। इंटेलेक्चुअल प्रॉपर्टी, इंटेलेक्चुअल प्रॉपर्टी की बात कर रहे हैं, मतलब आपके माइंड से जुड़ी हुई चीजें। मेरे सामने सर बैठे हुए हैं, उनके हाथ में कॉपी है, पेन है। मैं सर से पूछूंगी कि सर ये पेन मुझे चाहिए, सर ये नोटबुक मुझे चाहिए, मुझे परमिशन लेना होगा सर से। विदाउट परमिशन मैं उनसे कुछ भी नहीं ले सकती। तो इंटेलेक्चुअल प्रॉपर्टी राइट्स की बात मतलब लीगल प्रोटेक्शन ग्रांटेड फॉर इंटेलेक्चुअल प्रॉपर्टीज की बात होती है, जिसमें हम इंटेलेक्चुअल मीन्स आप क्या सोचते हो, थिंकिंग क्या है आपकी, इमेजिनेशंस क्या है, इनोवेशंस क्या है। इंटेलेक्चुअल प्रॉपर्टीज की बात करते हैं, हम माइंड में सोचते हैं कि अभी मुझे कुछ लिखना है, अभी मुझे कुछ आपके दिमाग में चल रहा है, कोई मॉडल बनाने का, कोई मशीन बनाने का, आप उस चीज को सोचते हो। बट वो सारी चीज जब आप सोच के उसके मैकेनिज्म्स को बना लेते हो, तो वह इंटेलेक्चुअल प्रॉपर्टी राइट्स होता है। प्रॉपर्टीज टू टाइप्स की होती हैं, टेंजिबल और इनटेंजिबल। टेंजिबल मीन्स जिसको आप सामने से देख रहे हो, आप जिसको टच कर सकते हो। जैसे आपके सामने एसी लगा हुआ है, दिस इज टेंजिबल। इनटेंजिबल क्या होगा इसमें? इसमें इनटेंजिबल क्या होगा? इसके अंदर का जो मैकेनिज्म होगा, इसके अंदर जिस मैकेनिज्म पे ये वर्क कर रहा होगा, वो इनटेंजिबल होगा। टेंजिबल प्रॉपर्टीज जो रहती हैं वो हमेशा मूवेबल होती हैं। यदि आज आपको कोई प्रॉपर्टी लेनी है तो आपको उसके लिए क्या प्रोसेस करना होता है? यदि आज आपको कोई घर लेना है, उसके लिए आपको क्या प्रोसेस करना होता है? आप उसके लिए प्रॉपर ऑफिशियली जिसकी प्रॉपर्टी है उसके साथ आप एग्रीमेंट साइन करेंगे। आप उनके साथ स्टाम्प पेपर में आपको सारा स्टाम्प पेपर लेना होगा। आपको जो गवर्नमेंट की फीस होगी उसके थ्रू आपको रजिस्ट्रेशंस कराना होगा। ठीक इसी तरीके से हमारी भी जो इंटेलेक्चुअल प्रॉपर्टी राइट्स होती हैं उनको भी हमको इसी तरीके से लेना होता है। तो इनटेंजिबल मींस इंटेलेक्चुअल प्रॉपर्टी की बात हो रही है, जिसमें कॉपीराइट, इंडस्ट्रियल प्रॉपर्टी रिलेटेड बात होती है, पेटेंट, डिजाइन, ट्रेडमार्क की। एक छोटा सा वीडियो आपके सामने। स्टूडेंट्स लोगों से पूछना चाहूंगी, इस वीडियो को देख के आप क्या समझ पा रहे हो? सॉरी, वो आपने देखा ये वीडियो छोटा सा? चलिए कोई बात नहीं। इट्स ओके। इसके जस्ट एक पहले स्लाइड थी जिसमें हम लोग नेशनल आईपीआर की बात कर रहे हैं इंडिया में, जो 2016 में लॉन्च हुआ, जिसमें ये बोला गया कि हम इंडियंस अपने कंट्री के इनोवेशन में अपना हाथ आगे बढ़ाएं। हम बहुत डिपेंडेंट हैं दूसरी कंट्रीज पे, जिसको लेके ये नई पॉलिसी बनी। तो व्हाई डू आईपीआर एग्जिस्ट? क्यों आईपीआर क्यों चाहिए? इंटेलेक्चुअल प्रॉपर्टी राइट्स की बात हम क्यों कर रहे हैं? एक्चुअली आईपीआर इंश्योर दैट आइडियाज आर प्रोटेक्टेड। आपके आइडियाज हमेशा सेफ रहेंगे। आप उसको क्रिएट कर रहे हो, आपको उसको रिवॉर्ड करोगे और आप उसको अपने इनोवेशंस को अपने लाइफ टाइम आप उसको कंटिन्यू करिए। दिस इज इंपॉर्टेंट। These are the 5 pillars of IPR. फर्स्ट, पेटेंट की बात करते हैं हम। व्हाट इज पेटेंट? पेटेंट दो तरीके से होता है, एक यूटिलिटी पेटेंट और एक डिजाइन पेटेंट। यूटिलिटी पेटेंट, यदि आपने सपोज नैनो मटेरियल लेकर आप रिसर्च कर रहे हो और उस नैनो मटेरियल के थ्रू आपने कोई लेटेस्ट डिवाइस बनाई है, उस डिवाइस के थ्रू आप एलईडीज पे जा रहे हो, कोई थिन फिल्म फैब्रिकेट कर रहे हो, उसका एक्यूरेसी क्या है, उसका पावर क्या है, उन सारी चीजों को आप, यदि आपने कोई नई चीज बनाई है तो आप उस पे पेटेंट कर सकते हो। यूटिलिटी पेटेंट पे आप जा सकते हो, ग्रांट पे जा सकते हो। और डिजाइन पेटेंट, इट्स वेरी सिंपल, बहुत सिंपल होता है ये। आप कोई भी चीज डिजाइन पेटेंट की बात करते हैं, यदि हम अपने इंडियन कल्चर की बात करें तो पहले जब हम चाय पिया करते थे तो मिट्टी के कुल्हड़ में पीते थे। मिट्टी का कुल्हड़ टूटने लगा तो हम मेटल्स में आ गए, मेटल्स आया, बोरोसिल आया, कांच की चीजें आई। क्यों? ज्यादा गर्म क्या करेगा वो? यस, है ना। तो उसके बाद आप कप बना, कप में एक साइड हैंडल रहा, आपको और इजी करना है, आपने दोनों साइड हैंडल्स लगाए। ये सारी चीजें जो है वो डिजाइन्स की बात कर। और यूटिलिटी पेटेंट की बात करते हैं हम, जिसमें आप कुछ नया सोच के, नई कोई चीज बना के उसको प्रैक्टिकली कमर्शियली आप कहीं यूज करवा पाते हैं। सेकंड, ट्रेडमार्क की बात करते हैं। ट्रेडमार्क मींस ब्रांड नेम, लोगो। सपोज आपका कोई छोटा सा एरिया है, आप उसमें एक छोटा सा ढाबा चला रहे हो, उस ढाबा का आपको नाम रखना है। आपने नाम रख दिया AS। उस नाम को, सपोज आप उसको AS नाम से आप ऐसे रन नहीं कर सकते। आप यदि उसका प्रॉपर, आप उसको ब्रांड नेम के लिए आपको जाना पड़ेगा, ट्रेडमार्क करना होगा, फिर आप उसको कभी भी यूज कर सकते हो। कॉपीराइट की बात करते हैं। अभी 19 फरवरी से 22 फरवरी तक साइंस कॉलेज में, साइंस कॉलेज ग्राउंड में एक बहुत बड़ा फंक्शन चल रहा था। किसी को याद है कुछ? हाँ, एक प्ले चल रहा था, नाटक चल रहा था। वो प्ले भी कॉपीराइट है। ये महाराष्ट्र का एक प्ले है, जाना राजा, और ये महाराष्ट्र शिवाजी पे बेस्ड है वो, और वो प्ले भी कॉपीराइट है। मतलब आपके अंदर जितना कुछ लिटरेसी से रिलेटेड, आप कोई बुक लिख रहे हो, आप कोई कविता लिख रहे हो, वो सारी चीजें आपके कॉपीराइट्स में आएंगी। व्हाट इज अ यूटिलिटी पेटेंट? मैं चाहूंगी कि बच्चे इंटरेक्ट करें, क्योंकि ऐसे ही बहुत कम लोग बैठे हुए हैं और एक साइड से सुन रहे हैं, तो बहुत लोगों को नींद भी आ रही है। तो थोड़ा सा आप बताइए मुझे कि यूटिलिटी पेटेंट के बारे में क्या है। आईआईसी आप जानते हैं? आईआईसी आपके कॉलेज में, आपके यूनिवर्सिटी में आईआईसी सेल होगा? आईआईसी सेल, यदि आप इससे अवेयर नहीं हैं तो हम पेटेंट की बात कैसे करें? इंस्टीट्यूट इनोवेशन काउंसिल सेल, यदि आप इससे अवेयर नहीं हैं तो हम कैसे पहुंच पाएंगे विकसित भारत की ओर? हम कैसे क्रिएटिव इंडिया बनाएंगे? हम कैसे इंडिया में नया नया इनोवेशन करेंगे? हम कैसे पेटेंट कराएंगे? पेटेंट के लिए जो प्रोसेस होता है वो सिंपल, आपके माइंड में कोई चीज आई, आप उसकी ड्राफ्टिंग किए, ड्राफ्ट एप्लीकेशन आपका जाएगा, आप उसको पेटेंट ऑफिस जो इंडिया में 4 जगह है, दिल्ली, मुंबई, कलकत्ता और चेन्नई में, वहां पे आप प्रॉपरली इसको पेटेंट ऑफिस में फाइल करते हो। आपका एग्जामिनेशन होगा, और एग्जामिनेशन मीन्स आजकल ऑनलाइन 1 आवर का सेशन होता है आपका, जिसमें प्रॉपर पूछा जाता है कि ये डिजाइन क्यों, ये मटेरियल क्यों, तो ये सारी चीजों को लेके इस पे काम किया जाता है। ये वेल नोन यूटिलिटी पेटेंट्स हैं जो ऑलरेडी आप इनके बारे में जानते हैं। सारा कुछ डायग्राम से आपको क्लियर हो रहा है कि यदि प्रिंटिंग प्रेस की बात कर रहे हैं,
Last 5 minutes.
यदि हम प्रिंटिंग प्रेस की बात कर रहे हैं तो आपको सारे नाम पता है, यहाँ दिख रहा है। डिजाइन पेटेंट्स में आप देखिए, ज्वेलरी भी डिजाइन पेटेंट्स में आता है। आप कोई चीज प्लान करके करते हैं तो ये डिजाइन पेटेंट्स की बात होती है। यहाँ पे आपको शूज दिख रहा है, बॉटल्स दिख रहे हैं, ड्रेसेस दिख रहे हैं, ये सारा कुछ आप डिजाइन कर सकते हो। आपको ये पता होना चाहिए कि 90% पेटेंट, आपके जो डिजाइन पेटेंट्स हैं उसको आप आगे कंटिन्यू कर सकते हो, उसमें आप और वर्क कर सकते हो, और वर्क करके उसको आप यूटिलिटी पेटेंट के लिए जा सकते हो। ट्रेडमार्क, जनरली ब्रांड और लोगो की बात होती है। यहाँ बहुत सारे ट्रेडमार्क, जैसे आप याहू, एडिडास, गूगल, ये सारी चीजें आप जान जाते हो किससे? ट्रेडमार्क के थ्रू। स्मेल, साउंड, वॉइस, 3D साइंस, ये सारा भी ट्रेडमार्क्स के थ्रू है। आइकॉनिक इंडियन ट्रेडमार्क्स आप जानते हो, ये 2.5 किलो का हाथ जब किसी पे पड़ता है तो आदमी उठता नहीं, उठ जाता है। ये सारा चीज भी आपका इंडियन ट्रेडमार्क्स में आता है। आपके सामने कपिल शर्मा जी का ये दिख रहा है, ये भी आप जानते हो, आप इसको यूज नहीं कर सकते। कुछ फेक, लीगल और इलीगल, सब में जो आपको पहले दिख रहा है, पहला वाला सिंबल किसका है? पेप्सी का। जो बाद वाला है वो फेक है। वैसे ही एडिडास का दिख रहा है आपको, इंस्टाग्राम का दिख रहा है, BMW का दिख रहा है। यदि आप इलीगली कुछ भी ऐसा करते हैं तो इसके लिए काफी हैवी पनिशमेंट आपको मिलता है। कॉपीराइट्स के लिए यहाँ पे बहुत सारी चीजें लिखी हुई हैं, आप दिख रहा है आपको। ये ओबामा का इलेक्शन के टाइम 2008 में ये पिक्चर यूज हुआ था। तो इसके लिए भी काफी कुछ कॉपीराइट्स को लेके इसके अगेंस्ट कोर्ट तक बात गई और काफी बड़ा पनिशमेंट मिला। उसके बाद फिर ये मामला ये हुआ। मतलब आप कोई भी चीज को किसी का भी इस तरीके से कभी भी यूज नहीं कर सकते। जीआई मींस ज्योग्राफिकल की बात करते हैं हम, इसमें भी आप जानते हो दार्जिलिंग का जो चाय है, कांचीपुरम की सिल्क साड़ी, कोल्हापुरी चप्पल, बासमती राइस, तिरुपति लड्डू, नागपुर ऑरेंज, ये सारा भी क्या है? आप इंटरेक्ट नहीं करेंगे तो यस, तो ये सारे आइटम्स भी आप देख रहे हो कि आप इसको ज्योग्राफिकल ओरिजिन से वो आया हुआ है तो हम उसको वैसा यूज नहीं करते। ये बॉटल की शेप्स आप देख रहे हो कोका कोला की, यहाँ पे आपको बताया हुआ है कि पेटेंट जो होता है, यूटिलिटी पेटेंट मैक्सिमम 20 इयर्स तक के लिए, ट्रेडमार्क लाइफ लॉन्ग होता है। डिजाइन आप 10 to 15 ईयर्स का रहता है, 10 ईयर्स का रहता है, फिर आप उसको 5 ईयर्स के लिए और एक्सटेंड कर सकते हैं। कॉपीराइट 60 ईयर्स के लिए रहता है और ज्योग्राफिकल इंडिकेशंस जो है वो लाइफ लॉन्ग की बात होती है। ये एक बहुत अच्छा वीडियो है जो हम लोग बात कर रहे थे कि इलेक्ट्रिक व्हीकल्स 2000 में भी आया, बट अभी क्यों इतना पॉपुलर हुआ, ये आप देखिए। कहाँ चले? हाँ, वर्ल्ड इंटेलेक्चुअल प्रॉपर्टी राइट ऑर्गेनाइजेशन ने ये एक वीडियो अपने साइट पे अपलोड किया है और बच्चों को ये सोचने के लिए मजबूर किया कि 2000 में भी जब हम लोग बात कर रहे थे उस टाइम पे भी इलेक्ट्रिकल व्हीकल्स की बात हो रही थी, बट अभी इलेक्ट्रिकल व्हीकल्स इतने सक्सेस कैसे हो गए, ऐसे कैसे बैटरी की बात आ गई। मेरा जो कंट्रीब्यूशन है यूटिलिटी पेटेंट्स में वो 2 है। हम एक ऑर्गेनाइजेशंस के साथ SSIPMT शंकराचार्य के साथ एक स्टूडेंट्स के साथ हम काम करते हैं। उस स्टूडेंट्स ने ये प्ले नहीं हो रहा है, इसमें साउंड नहीं है। ये ज्योमेट्रिकल बॉक्स के लिए हमने पेटेंट किया हुआ था जो हमें ग्रांट इसमें मिला है 20 ईयर्स का। आप जो पहले ट्रेडिशनली जो ज्योमेट्री बॉक्स आती थी उस ज्योमेट्री बॉक्स में आप स्केल से लाइन खींचते हैं, प्रोटेक्टर से एंगल्स बनाते हैं, राउंडर से सर्कल बनाते हैं। ये जो डिवाइस है ये ऑलरेडी इस पे काम कर रहा है। इसमें साउंड नहीं है, इसमें इसके पूरे मैकेनिज्म्स को मतलब बताया गया है, स्पेशली बोल के कि किस मैकेनिज्म पे कैसे ऑर्डिनो के थ्रू ये काम कर रहा है और कैसे इसको हमने बनाया है। These are the references. Thank you so much.
Thank you so much, Dr. Shobha, for your wonderful talk on IPR. I think we need to get an idea for our new technologies to start startups. Like, suppose you want to establish your business, then first of all, you need to get a utility patent for that. It is nothing but a government document to which we will have a license for that. Okay, thank you so much. So, students, if there any doubt, please ask.
चलो कोई, हाँ बोलिए।
मैम, ये जितने भी पेटेंटिंग की आपने बातें बताई, इट्स वेरी नॉलेजेबल, लेकिन मुझे एक डाउट है जो मैं पूछना चाहती हूँ। मैम, ये हम जो पेटेंट कराते हैं जनरली इंडियन पेटेंट्स पे जाते हैं, पेटेंट हम करते हैं। तो ये जो ऑस्ट्रेलियन पेटेंट होता है, यूके पेटेंट होता है, हमारे ये जो इंडियन पेटेंट होता है, इसमें क्या फर्क है और हमको क्या चूज करना चाहिए?
यदि हम इंडियन नॉलेज सिस्टम की आज बात कर रहे हैं, हम विकसित भारत की बात कर रहे हैं, तो मैं तो भारत की तरफ ही जाऊंगी, तो मैं इंडियन पेटेंट्स की ही बात करूंगी। और हमारे सेशन चेयर आपको धीरेन्द्र सर बताएंगे कि ऑस्ट्रेलियन पेटेंट और ये सारे पेटेंट्स पे हमको क्यों नहीं जाना चाहिए।
यूएस का अच्छा लेवल मार्केट है, पर दूसरा सेकंड वन इज इंडिया। बाकी देखिए जो दूसरे कंट्रीज जो पेटेंट्स दे रहे हैं आपको, जैसे ऑस्ट्रेलिया हो गया, यूके हो गया, वो ना डिजाइन पेटेंट्स दे रहे हैं और इसका एक एक्चुअली प्रोसेस है। नाउ हम देखते हैं उनके मैसेजेस आता है, उसका एक बिजनेस चल रहा है। कि उसमें यूटिलिटी पेटेंट्स के बारे में किसी को आइडिया नहीं होगा। वो लोग क्या करते हैं, आपको जैसे फर्स्ट पोजीशन, सेकंड पोजीशन, थर्ड पोजीशन, अब फ्रीक्वेंट में आपको मिल रहा होगा 5,000, 10,000, 20,000 ऐसा, वो पब्लिश करा के छोड़ देते हैं।
कोई बैठा है इंग्लिश बोलने से बच्चे सुनते हैं।
पेटेंट फाइलिंग, और वो भी अमेंडमेंट हो जाता है उसमें। तो आप जब भी चाहें ना तो इंडियन पेटेंट
अब बच्चों को आईएलटीएस का फुल फॉर्म नहीं
और
ये अपने को दिख यही जानना है।
ग्रांट में आप फास्ट एक्सप्रेस एग्जामिनेशन से जाते हैं ना, तो आपको जैसे अभी लेटेस्ट में हमने पेटेंट किया है 4 बच्चों के साथ, वो 6 मंथ और 22 दिन में 22 डेज में, अदरवाइज नॉर्मल पे जाएंगे तो 2 to 3 years में। तो ये एक बड़ा बहुत बड़ा फील्ड है। इट्स मतलब लाइक नॉट लेस देन ओशन लाइक दैट।
एक्चुअली पेटेंटिंग के लिए मैं सभी को एक इन्फॉर्मेशन देना चाहती हूँ। मैं पेटेंट करती हूँ, करवाती हूँ, अपने से ही फाइल करती हूँ, इसलिए मैं रेस्ट ऑफ द फैकल्टीज को भी इन्फॉर्मेशन देना चाहूंगी कि पेटेंट हम मात्र 1600 रुपए में भी 1800 रुपए में भी कर सकते हैं। 1600 रुपए में 2 टाइप के पब्लिकेशंस होते हैं, एक होता है अर्ली पब्लिकेशन, एक होता है लेट पब्लिकेशन। अगर आप अर्ली पब्लिकेशन को फाइल करेंगे तो
थोड़ा मतलब हड़बड़ हो जाता है।
नॉर्मली 4200 रुपए ही होता है, उसमें आपको 1 महीने के अंदर आ जाता है। और अगर आप जो लेट पब्लिकेशन होता है,
अपन निश्चित भारत की बात कर रहे हैं, इंडिया में प्रोडक्ट बने, होली में बात कर रहे हैं, तो भारत के बच्चे
टाइम लगाता है, 1-2 साल का बहुत टाइम, 18 मंथ भी लगा सकता है। तो पेटेंटिंग अच्छा बहुत ज्यादा इजी, शुरू से इजी प्रोसेस है, लेकिन बस इन्फॉर्मेशन की कमी है। तो अगर आप कोई पेटेंट करना चाहें तो आप लोग मुझे इन्फॉर्म कर सकते हैं, मुझसे कॉन्टेक्ट भी कर सकते हैं। थैंक यू। Thank you. We sincerely thank you for taking your time to share your thoughtful view and valuable perspective with us. Thank you so much, ma'am. And now may I request Dr. Shilpa Sharma, ma'am, Dean Science, to felicitate our speaker, Dr. Shubha Mishra, ma'am, with memento kit and certificate as a token of appreciation. Can we have a huge round of applause?
वापस रखिएगा और यूनिवर्सिटी टिकट। पेज में फोटो वीडियो बनाएंगे। ओह
And for the sequel, who are not physically present, our next speaker is here with us online. Let's welcome Dr. Shama Bagman, who is joining us virtually today. Despite being injured, has taken the effort to join us today. Thank you, ma'am. We truly wish you a speedy recovery. Let's welcome her with a big round of applause. She is joining virtually today with us. I now invite Ayushi to take over for the introduction. We now move to our next speaker, Dr. Shama Bagman, whose journey as microbiologist started since 1991 while pursuing PG in life science. She has nearly 30 years of teaching experience in microbiology and botany as assistant professor in various colleges, universities of Chhattisgarh and Madhya Pradesh. She has graduated and pursued MSc and PhD from Pandit Ravi Shankar Shukla University, Raipur. She has lifetime membership in professional bodies, MBSI, MPCG, Darshan Parishad Advances in Life Science, Trends in Biosciences, Society for Science and Environment, ICPEP, Indian Journal of Ecology, and Indian Botanical Society, and membership in around 20 communities. Also, she has attended 50 above conferences, seminars, and 40 webinars
at national and international levels and presented papers. She has edited 23 books on her name, have written 32 book chapters, published, co-author in a conference souvenir, guest editor in journal volume, 2 abstract books, guest editor in research journal, and 2 monographs as her achievements. So I request Dr. Shama Bag ma'am to share her valuable thoughts with us through online. Am I audible, ma'am? Good afternoon, everyone.
Hello, good noon, ma'am. You are audible perfectly.
Okay, thank you so much. Good afternoon, everyone. Present, Honorable Vice Chancellor, distinguished guests, respected colleagues, researchers, and dear students, and I thank convener Dr. Rajesh Kumar and Dr. Jayakumar Devangan, organizing secretaries Dr. Shilpa Sharma, Dr. Sachin Kumar Das, and all the organizing committee members for inviting me as a speaker. Today, I am privileged to speak at this international conference on global convergence, business, technology, and humanities for sustainable development. Glad to say that the theme is not only timely, but it is an urgent thing that we should think over it. And we are living in a defining century of human history. On one hand, we witness unprecedented technological advancement such as artificial intelligence, biotechnology, digital transformation. But at the other hand, we face climate change, biodiversity, collapse, water scarcity, and deepening social inequalities. Shall I present my slides?
Yes, please
And
proceed.
share the screen. Yes, okay.
इससे तो आवाज आ रहा है ये तो आवाज नहीं आएगा ना
Is it visible?
Yes, ma'am, visible.
Okay. So I'll be talking on reimagining sustainability for a resilient future. आज के समय का बहुत ही कहना चाहिए एक ऐसा टॉपिक जिसपे कि हम सबको सोचना चाहिए, विचार करना चाहिए और ये विचार करते करते हमारे उसमें हमने बहुत समय निकाल दिया। अब हमारे को काम करना है इस पे ताकि हम 2030 की हमारा गोल है सस्टेनेबल गोल वो फुलफिल कर सके। Is it moving, ma'am? Are my slides moving?
No, ma'am. Yes, now it is.
Are my slides moving?
Yes, now. Yes, fine, ma'am. Please proceed.
So this is a picture where we can say that it's an imagining alternative futures in the Anthropocene. That human development which has led to this much of the pollution which we are seeing here and which is from this greenery we have emerged to
towards this pollution. We have the goals which are known as the Sustainable Development Goals, which had 5 pillars, and we have to complete it by 2030. And our world will be, we are saying that we will achieve the Sustainable Developmental Goals, which are 17 in number, and which is having these 5 pillars: the people, the planet, the prosperity, the peace, and the partnership. And this goal can be done, and my presentation explores the vital role of all of us, we are all the environmental humanities, in addressing this complex challenge of the Anthropocene by integrating diverse knowledge systems which will arise from the scientific data to indigenous wisdom, and from artistic expression to the philosophical inquiry, we can cultivate a more nuanced understanding of our relationship with the planet, the only blue planet or the green planet, we can say. Join us as we envision a future where sustainability is not merely a technical problem, but a deeply cultural, ethical, and collaborative endeavor, as said by one of the environmentalists. So, why we are thinking? Why the humanities matter? Why this is now thought? Because it is something more than science. The current approach or the geological approach is marked by significant human impact on Earth's geology as well as ecosystem. And it requires a holistic approach. All of us, we have to join the hands. Alone, scientific data which is on the papers cannot do anything. We have to address the cultural, ethical, and the emotional dimension of this environmental crisis. Why this is happening? How much we are responsible? Or 100% we are the one who are responsible for this. Environmental humanities offer frameworks to understand the deeper societal roots of these challenges. Now, what can we do? We can connect our diverse knowledge system. Environmental humanities acts as a bridge linking history, philosophy, literature, art, and indigenous knowledge systems. We have to inculcate all this into one platform and work together. And this is the only thing which can lead to understand the human nature interaction. From purely scientific or economic perspective, we have to incorporate our lively experiences, our spiritual connections, and our long-standing cultural practices that offer profound insights into the sustainability. And we have to work together along with these knowledge systems, and this will act as a bridge to gain this aim. How can we do that? Stanford Sustain 140 is running a course which exemplifies this integrated approach, and they are engaging the students in critical reflection on environmental issues through diverse lenses. Each one is seeing the nature by their own eye and doing the best to protect it. And these programs cultivate not only a deeper understanding, but also inspire creative and ethical solutions. Here, I want to say that we have also added the environmental sciences in our courses, but we are not looking into that mirror. We are what we are doing. We are just taking the classes and giving the marks to the students for the environmental sciences. No, we have to equip all the individuals to work for the environment, to cure the environment, and to have a sense of responsibility over it. What is the crisis beyond the science? What are the cultural and the ethical roots? How much destruction we have done? The degradation of environment is not a technical problem. It is fundamentally a crisis of meaning, values, and human relationship with the natural world. We are away from the nature. We just don't want to live with the nature. We just want to live in the AI world. So we have to change ourselves. What we do, we prevail or prioritize the economic growth and technological solutions, and we overlook our cultural and the spiritual bonds which many communities share with the environments, and they protect the nature. This oversight leads to a detachment from the emotional and the psychological impacts of environmental change, making it harder to foster collective action and genuine care for the planet. ये जो सिद्धांत है प्लानेटरी बाउंड्रीज का, ये हमको बताता है कि पृथ्वी की भी अपनी सीमाएं हैं। यदि हम इन सीमाओं का अतिक्रमण करते हैं तो पारिस्थितिकी संतुलन जो है वो बिगड़ जाता है। और जो सतत विकास का अर्थ जो होते हैं, जो हम ग्रोथ बोलते हैं, जो ग्रोथ बोलते हैं वो केवल आर्थिक वृद्धि नहीं है, इकोनॉमिक ग्रोथ नहीं है। ओनली द इकोनॉमिक और टेक्नोलॉजिकल ग्रोथ इज नॉट अ विकास, you know. It includes the growth as well as the पर्यावरण संरक्षण or the environmental sustainability plus सोशल डिस्क्रिमिनेशन जो है उसको रिमूव करके हमको इन सबको एक साथ ले करके हम एक प्लेटफॉर्म पे हमको चलना होगा, और ग्लोबल कन्वर्जेंस से बिजनेस, टेक्नोलॉजी, ह्यूमैनिटी इन सबको एक साथ जोड़ना होगा। और एनवायरमेंटल ह्यूमैनिटीज में ह्यूमन बीइंग्स की जो पोजीशन है वो हमें मेन स्ट्रीम से जो मार्जिनलाइज करती है उस व्यू को हमको इंडिजिनस कल्चर के साथ में अटैच करना होगा। देन ओनली वी कैन डू दैट, एंड दीस डोमिनेंट नैरेटिव्स कैन inadvertently perpetuate environmental injustices by ignoring local knowledge and the disproportionate emotional burden placed on the vulnerable communities. हम challenge क्या देते हैं? We say that they will do that. No, we should say that we will do that, or I will do that. As he's in 2016 argued that the environmental humanities compel us to critically examine and reframe what it means to be human on a rapidly changing planet, urging us to question the assumption that underpin our current ecological trajectory. कैसे पहुंचेंगे हम वहां तक, किस तरह से हमको मूव करना है? तो जो ग्लोबल कन्वर्जेंस, नाउ ग्लोबल कन्वर्जेंस से जाने के लिए हमको पहले हमारे घर से शुरू करना होगा, एंड दैट इज द ब्रिजिंग द नॉलेज सिस्टम, दैट इज ह्यूमैनिटीज फॉर द एनवायरनमेंट. We have to collaborate with strengthening networks by emphasizing inclusivity, and UNESCO bridges all these, and this coalition serves as prime example of international cooperation driven by humanities, demonstrating that approach advanced global sustainable growth. And these diverse epistemologies, which on ground level, local context and traditional wisdom has to be inculcated in that, and this developing equitable and cultural motive. How we value the humanity
Excuse me, ma'am.
beyond the economic metrics. अब हमको economic metrics को छोड़ना होगा.
Ma'am, sorry to interrupt you. Slides are not moving ahead.
Okay, sir.
We are on.
Right to me.
Right now.
Okay.
Yes.
Is it now?
Yes, ma'am.
Is it now moving, sir?
Yes, ma'am. It is now.
Okay, sorry for that. Let me show the previous slides also. Just I'll be moving the slides. This one, then I told about this one, and then this one. Okay. Now is it visible, sir?
Yes, ma'am.
Okay. Now the valuing plurality beyond economic methods. Beyond economic systems, we have to do the relational and ecocentric valuations we have to do. What we have to do, environmental humanities, it advocates for relational and ecocentric valuation methods. What are these ecocentric valuation methods? We have to start from ourselves. I always say that between all living and non-living components of an ecosystem, we have to prioritize the indigenous and the local knowledge. They are very good at the nature. They know how to preserve the land, the water, and the biodiversity, and that we have to inculcate ourselves with them. What thinking we should have, or what thinking we should allow to our youngsters, what we should do? We should, for inclusive future thinking, tools such as participatory mapping, community-led narratives, and speculative imaginaries. Now, what we do when we are told that we have to prevent the nature, what we do, we give students a course which is known as the environmental science, which they never take it seriously. They never go for go to the environment and do the activities and save the environment. They only read it and बाकी का काम तो क्या है जो लोग कर रहे हैं ना दूसरे लोग जो बचा रहे हैं धरती बचाओ वो करेंगे हम तो कुछ नहीं करेंगे हम केवल पढ़ेंगे और हम आगे बढ़ेंगे नहीं हमको ये नहीं करना है हमको इसको theoretical से बनाना है हमको इसको practical बनाना है this environment value should be given as a practical knowledge इसको हमको practically करना है फिर हमने एक लेक्चर करा लिया एक हमने जिस दिन हमारा वर्ल्ड एनवायरनमेंट डे हो गया थोड़ा पौधे लगा दिए थोड़ा हमने साफ सफाई कर दी प्लास्टिक उठा के दिन दिया थोड़ा एक जगह फोटोग्राफ्स कर दिए और हमने सोशल मीडिया में डाल दिया दैट शुड नॉट बी डन सो हमको जो फ्यूचर थिंकिंग करनी है वो ये करना है कि हमको ये सारी चीजों को एक प्लेटफॉर्म पे लाकर के उससे वर्क करना है एंड द बेस्ट प्लेटफॉर्म आई थिंक इज द students platform where we can do that and हमको उनमें वो inculcate करना है जिससे कि हम एक इकाई में अगर ये चीज डालते जाएंगे तो वो आगे हमारे नेचर को बहुत ज्यादा हेल्प करेगी Alternative futures in the anthropocene हमको क्या करना है जो सोशल एडिक्शंस है वो ब्रेक करना है इसको मैं ज्यादा डीपली नहीं दूंगी हम सब जानते हैं कि हमारा एडिक्शन कहाँ पर है उस एडिक्शन को जब तक हम रिमूव नहीं करेंगे आगे नहीं बढ़ सकते हैं we have to overcome the barriers we have to engage the wider publics from class 1 we have to start this है ना some case studies I have taken when I am having time or I have to move faster
Ma'am, excuse me. Sorry to interrupt you in between again. We are having last 3 minutes to finish presentation.
yes
We are having last 3 minutes, ma'am, to finish presentation.
Okay. These are some of the case studies which are environmental humanities course, interdisciplinary integration, holistic understanding, community engagement projects they have to do, fostering relational ways of knowing, and motivating collective responsibility. I have collected some of the points that how we can do that. We can shape the environmental ethics by cultural works as works as a catalyst. In films, in poetry, in literature, and visual arts, we can shape the environmental, ethical, and the public imagination. Emotional connection we have to develop between the scientific reports and the human and the society. Bridging the science and the experience. How can we give to the society by storytelling or by creating engagement like catalyst, articulist, planet Earth arts, and these type of little small things. And these powerful mediums, we can inspire a generation to imagine and create a more sustainable world. And towards a resilient future, major takeaways क्या हो सकते हैं? Global convergence of knowledge and values, reimagining human nature relationships, embracing plurality, justice, and creativity. What Globally, what we are doing, we have research institutes and networks involved which are working on it. We have some human impact data which are telling us that how many documented human impacts are there. Fisher et al. Has done a great work over that, and we are tracking anthropogenic emissions since 2006, and we have to publish the methodologies and the outcomes for sustainable future. Next, we have to join the movement to reimagine the sustainability. We have to embrace interdisciplinary dialogue. We have to foster the ethical reflection, and we have to build a just resilient world. Finding our place on a changing planet, it starts with rethinking who we are and how we live. Our engagement, commitment are vital, shaping our future, sustainability, and it is not just a goal, but a lived reality for all of us. So, in the end, we have to attain these all sustainable goals till 2030, and we are working together, and we can achieve them if we join together. And thanks to all of you for this opportunity to me, providing this opportunity to me. And these sustainable goals, we say, to conclude that sustainability is not only a scientific framework, it is a moral responsibility of all of us. We have to rethink our relationship with nature. When business, innovation, technological advancement, humanistic values converge, we can imagine and create alternative future. A resilient future is possible, but it requires collective action. Together, we can achieve it. Thanks to the organizer for providing me those opportunities.
Thank you so much, ma'am. You have explained actually the things related to the humanities backgrounds. Okay, very wonderful talk. If there is any questions, please ask. Otherwise, we will move further. Okay, ma'am. Thank you once again.
Welcome, sir.
Thank you so much, Dr. Shamabraj ma'am, for taking the time to join us. Your presence, despite your injury, means a lot to us. We hope you recover very soon. And moving forward, now I would like to invite Dr. D.P. Chandrakar ma'am to welcome our esteemed speaker, Dr. Gulab Chand Arya sir, with a bouquet as a token of gratitude. And now I hand over the mic to Ayushi for the introduction. Next, we have Dr. Gulab Chand Arya sir, who is currently working as a Ramalinga Swamy Re-Entry Fellows at School of Biotechnology, KIIT University, Bhubaneswar. Prior to this role, he worked as a postdoctoral fellow at ARO Israel at the Department of Plant Sciences. With a research and academic career spanning over 8 years, his expertise lies in the field of plant surface pathogen interaction. Plant G protein signaling, Dr. Arya's research efforts have yielded substantial outcomes, resulting in the publication of over 19 research articles in reputable journals, along with paper presentation in over 10 national and international conferences and contributions to 3 book chapters. He serves as a reviewer and editor for numerous esteemed journals. Dr. Arya is lifetime member of prominent organizations including the Indian Society of Plant Physiology. His contributions have been recognized through notable awards. The DBT awarded him the Ramalinga Swamy Re-Entry Fellowship Award. So, I would like to invite Dr. Gulab Chand Arya sir on stage to share his valuable thoughts with us.
Hello. 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 Hello.
Mobile आसपास mobile.
Thank you. I continue. Hello. No. There's no sound. Hello. Hello. Can you hear me?
Yes.
Thank you, organizer of INCON 2026. I'm Dr. Gulab. Currently, I'm working as a Ramalinga Re-Entry Fellow and Assistant Professor in KT University, Bhubaneswar. This is my brief intro. So I did my PhD from National Institute of Plant Genome Research in JNU. And then I moved to Israel for my postdoctoral fellowship. I completed 5 years there, and then I got Ramalinga Re-Entry Fellowship. And I activated it in KT University, Bhubaneswar. So this is my research area. I'm working on plant surface pathogen interaction. So yeah, pointer here. So these are some of the pictures from my research work. I work in my past. I work on cucumber and tomato. Or currently in my lab, I'm working on foxtail millets. These are small millets. How different and different pathogens interact with these leaves of these millets. I'm going to share some story from my work. Two story I will share today from cucumber and tomato. So what is the plant surface? Like we humans have human skin, plant also have autonomous skin. We call it cuticle, and it is mainly composed of lipophilic barriers. So what are plant lipophilic barriers? These are plant protective vaxillary layer present in almost all land plants. There are two major type of plant lipophilic barrier. First is cuticle, which is present in most of the plant parts, including roots, stem, leaf, flowers, and fruit. So the shininess in the most of the fruit you see is because of this plant lipophilic barrier. Another one is suberin, which is present in the fruit surface of many fruits. You can see potato, melons, and cucumbers. And this is also present in root endodermis and in seed coat. If you see the structure of plant cuticle, it is present in the outermost layer in the epidermal cells, and above, this is mainly present above the cell wall. And above the cell wall, cuticle also contains waxes. We call them epicuticular waxes. So if we see the chemical nature of cuticle, it is mainly composed of cutin and waxes. Cutin mainly composed of different monomers, C16 and C fatty acids, C18 fatty acids, and there is different derivatives. And waxes are composed of C20 to C40 very long chain fatty acids. And if you talk about suberin, it is mainly composed of aliphatic as well as phenolic compounds. They are cross-linked together with the help of ester bonds. This plant cuticle and other lipophilic barriers play a very, very important role in almost all almost most of the things, like they provide mechanical support, they prevent organ fusion, they provide against UV radiation, they limit water loss, and one of the most important is they also restrict the pathogen entry. But somehow fungal pathogens and bacterial pathogens can degrade this cutin and cause many different diseases. In, like, this is example of tomato. And how they do it? So fungus or bacterial species, they secrete some specialized enzymes which can degrade this cutin and praxis. And once they degrade, they can penetrate inside and cause serious diseases. So we wanted to study the role of cuticle. What is the role of cuticle in tomato? And to study this, we use cutinases. So if you wanted to study for students, if you wanted to study function of any organ in plant, we have to disrupt it, overexpress it. So here we use a enzyme cutinase, which is secreted by fungal species, and then we specifically express this cutinase in tomato skin. If you use cutinase in tomato skin, it will degrade it, and we will try to understand what will happen if it is degraded. So what we did here, we choose very specific promoter which only express in the skin of tomato. We fuse this promoter with a cutinase enzyme, and then we make the transgenic plants in tomato. With the help of agrobacterium mediated genetic transformation, and we developed this transgenic plant. So you can see the first two lines, the upper one, first and second, are completely normal plant. They are control plant. And the second, third, and fourth are different lines we got once we generated the transgenic plant. So due to the effect of this cutinase enzyme, we found different phenotypes. So you can see the arrowhead is representing the disruption in the tomato skin. Other than this, we also find some other phenotypes like fruit length, fruit width, and fruit weights are also decreased. So we're surprised why this is happening. So our initial idea was, if you will take cutinase, it will degrade it. But here we saw something different. The skin was patchy and it was very hard. And we found this phenotype in many different lines we generated. It was very consistent. So analyze the skin of these fruits. So first and second are control plants, and you can see the third one is a mutant plant. You can see the difference between wild type. You can clearly count the cells. They are completely differentiated from each other. The cuticle is very integrated. But in the transgenic plant, you will not be able to count the cell. You will not be able to demarcate them. Further, we also took this skin of these fruits and we performed scanning electron microscope to see what is really happening in these fruits. So this is normal. First two are normal. In the third one, you can see there are the cells. The morphology of cells is disrupted. There were some bulges in the cells, and also there were some cracks, which is probably due to an expression of cutinase enzyme. Second, we can also make the sections of these fruits, and then we can stain with different dyes to see what is happening in the cuticle. You can see first and second are control, and third and fourth are transgenic plant. So the cuticle, the overall cuticle thickness was much increased in these plants. Our initial idea was, we will use cutinase, we will degrade it. If it is degraded, it should be thick. But here we got completely opposite result. So this is what happened. If you disturb any biological system, it's not necessary, but your initial idea may fulfill. So we got completely opposite results, and we also stained with different dyes. This is the typical symptoms of sugaring periderm formation. Sugaring periderm formation. And this is very common in many fruits, and this was also present in the bark of the trees. So we wanted to next see what is happening in the chemical nature of this cutin. So we found that cutin content was much increased especially in this transgenic fruit. So because of the degradation, plant somehow disturbed, the genetic mechanism is disturbed. Because of that, it's somehow boosting all the cutin content, and different monomers were also very high. When we did GCMS analysis to see what is happening in the gluten content. We also found something interesting. It was not previously reported because of the degradation of gluten. Gluten is it. We also found sugaring content. The plant take it as a stress, and to cope up with this stress, it started to accumulate sugaring synthesis, which is not a part, which is not normally present in the tomato fruits. Next, we also tested something is missing here, some figures. We also tested the waxes, the signiness I talked about. Signiness in the fruits is because of the waxes. Waxes was also very high. So to see why this is happening, why we try to degrade it, we try to make it thin, but here we got completely opposite result. Why this is happening? We performed transcriptome analysis to see what are the different genes responsible for this. So we found that cutin biosynthesis, wax biosynthesis, lipid transport, sugaring metabolism was very high. All the genes related to this pathway was very high. Other than this, cell wall biosynthesis, because cell critically is deposited above the cell wall. Cell wall biosynthesis genes was also very high. We also found different regulators like different milk transcription factor which regulate these pathways was also very high in these fruits. Next, we studied what is happening to the different physiological parameters like stiffness, quality of the fruit. So we tested roughness, elasticity of this plants, stiffness, how hard these are. So all these parameters were very high because of this accumulation of high gluten content, also high sugaring content. We also stained this seed, these fruits with the toluidine B dye to see what is happening to the permeability. The permeability increase was very high because of this gluten degradation. And this when we store this fruit for almost for a month, the fruit becomes very small. They shrink very fast and they appear like dried tomato. Next, we also tested whether these fruits can restrict the entry of pathogen. And this is very important to check the cuticle is disturbed, how it is response against different pathogens. So we checked it and it's very common for Patisseria fungus infecting tomato plants. The cuticle was thick. Because of the cuticle was thick, pathogen cannot enter inside the innermost flaccid tissues. And we also tested how spores behave. The spores of fungus can grow, but they are not unable to penetrate inside it. So here we have too much infection. Here is very minute infection in these tomatoes. So this was our final conclusion. If you disturb plant cuticle in tomato by expressing a cutinase, epidermal cells sense it somehow, it boosts many different genes to accumulate more cutin and waxes. Because of that, we found high cutin and high sugaring content, and the physiological effect was pathogen cannot enter inside these fruits. So this is one story. We already published this, and this is another story I'm telling you. Maybe you know this is cucumber, normal cucumber we eat, and this is another cucumber. Anyone maybe saw it? This was available in market from I think 10 to 20 years ago. This cucumber was very common. This cucumber, I ate in my childhood this cucumber, and it's very common in northeast part, especially in Sikkim, Nagaland. That is why we call it Sikkim cucumber. So there is a huge difference between these two fruits. It look like small, it look like very big, but there is a difference in the skin. So we wanted to see what is happening in this skin. So what we did, we performed microscopy again to see the cells, and similarly what I showed you just few slides back. In the Sikkim cucumber, there was a cracks, and in cracks there was something whitish. When we profile these with the help of this is histology. So when we perform histological sections, we found cuticle. The thickness of cuticle was much in Sikkim cucumber, and this is natural phenomena. We did not disturb anything here. This is completely normal, growing wild in nature. We also perform GCMS analysis to see what is happening in this skin. Similarly, what I showed you just results we got in tomato, we also found similar results in sativas, which is the normal cucumber we eat was completely less cutin, less waxes, but in Sikkim cucumber there was high wax and different aromatic fatty acids, other diacids, they were very, very high. So these were the results. We also tested this against pathogen and we found very, very interesting results. This normal cucumbers, what we eat in our home, they can be penetrated by this fungus very, very easily. But the other cucumber, Sikkim cucumber, they do not allow the entry of the pathogen. These are other results. The fungus can grow over the surface, but it cannot penetrate. Overall, the hyphal growth was high, but when we cut these fruits to see the infection, there was completely no infection in this. And the other fruit was completely infected. It got rotten within 3 to 4 days, but nothing happened to the Sikkim cucumber. So these were some articles we published in last 5 to 7 years. And this is my current lab, my PhD student, these are the master students who are working. And I'm working in plant surface pathogen infection. And if anyone would like to collaborate, I'll be happy. This is Kagai, my PI, and this is Ruth. Most of the studies related to NMR, stiffness, we did with Ruth. And we have funding from Department of Biotechnology. This Ramayana was a very entry fellow, almost about 1 crore for 5 years. And this is our School of Biotechnology at KT University. Thank you. Thank you very much.
Thank you, Dr. Aril, for this wonderful session. Is there any question from the student side? Okay, thank you once again.
Yes. Yes, sir.
You can use this one.
We sincerely thank you for sharing your knowledge with us. Now, may I request Dr. Rishi Jaiswal, Associate Professor, Department of Physics, to felicitate our speaker, Dr. Gulab Chand sir, with memento, kit, and certificate as a token of appreciation. And moving forward, now I would like to invite Dr. Sachin sir, Head of the Department, Microbiology and Biotechnology, to welcome our esteemed guest, Dr. Ashish Saraf sir, to welcome with a bouquet as a token of gratitude. And now I hand over the mic to Vegeta for the introduction.
Is everyone getting my voice? Is it okay? Fine. Thank you for
not so late. Okay, fine.
Our final invited speaker for this segment is Dr. Ashish Saraf sir, who is currently working as a professor and head at School of Science at University Raipur, Chhattisgarh. Prior to this role, he worked as assistant professor at the Department of Microbiology and Biotechnology, Mata Gujri Women's College, Jabalpur. He also served as a medical microbiologist and quality manager at the Department of Pathology and Microbiology, Anand Institute of Medical Science, Jabalpur, India. His involvement extends beyond research as he serves as a reviewer and editor for numerous esteemed journals. He also holds 5 patents to his name. So I request Dr. Ashish Saraf sir on this stage to share his valuable thoughts with us.
Thank you very much. Good evening, one and all. Life is very deciding, and we encountered by the hundreds of microbial challenges daily, regularly. We encountered by the daily, regularly, by the number of thousands of radiation, different kinds of radiation. But something is there that keeps us fitter and healthy, which is working behind the life, so it's working inside the body. And there is none other than the immune system. They keep us fitter and healthier. Reason, because it is having a great ability. The nature is providing that very good to him. We find balance a different kind of nature design. Dr. Rafi Saab, before you with presentation on biochemical characterizations of potential food excess, loaded nanoparticles for treatment of cancer. The major threat of life, which completely out of the control to treat. And it is very difficult to be recognized at its first stage. That is the treatment. Here is the content of the work, general introduction, we discuss a bit, review literature, methodology, findings and discussions I had merged together. So the cancer is described as a medical condition when the cell of our body is disobeyed, disregard the regular way of proliferations and multiplications, then they get to be uncontrolled. They are differentiated by this own need and eventually cause the invasions into the neighboring tissue. That is called metastasis. Basically, metastasis is the conditions. It is the conditions when the cancerous cells or the myeloma cells or the malignant cells is spreading in the different part of the body through the fluid connective tissue. And that is none other than the lymph and blood. The cancer is the primary cause of the death in many cases, including the early detections, old age, being of immune systems in the old age, existence of hazardous chemicals, and of course, the accumulations of the different kind of the drugs in the body, diagnosis procedure, lead diagnosis, and all. Now, here is the different kind. Here is the okay. Now, here is the different types of the cancers. The one is colorectal cancer. It's the third most largest worldwide. It is occurring in the worldwide about 1.8 million people yearly. The most common is lung cancer, breast cancer, and this is the percentage of incidence occurs worldwide yearly. About 10% overall reported cancers, the 13% of lungs and breast cancer, as far as the 10% is colorectal. So we get the great vaccines till the time, so that the percentage of mortality rate is quite low in the past 5 years, particularly in breast cancer.
cancer is the 100% mortality. It's a life threat. So it is a medical condition, basically. The cancer is a medical condition. When occurs, the group of cells is discarded in natural signal transmissions of the proliferations and cell differentiation. The cause of cancer is mostly the aging, failing of immune systems, bacteria and viruses. The mutations in the gene is caused. It also includes the genetic and history, the environmental factors and toxins, defective DNA repair, because the DNA is regularly damaged by the radiations. We don't understand how precisely it is damaging, but it is our body system that's automatically repaired. It's called a point mutation. But any kind of the deletion or additions of a single amino acid is there, it ultimately leads to a form of the cancer. And undoubtedly, the non-coding DNA, the non-coding DNA are very, very expressive. Most of the genes are in this dormant state, the non-coding part of the DNA. Only the 6% DNA will transcribe in whole life and they express whether the color of hairs or skin or eyes or anything. As far as rest of the 95% never ever express. And as far as the non-coding reason, forget it about it. Our moms, our Rishi Muni, they still in their life, they are just reviving and imbibing those genes which is encoded in the non-coding region of the DNA. Forget it. So types of treatment if we talk about, the major and the last treatment is surgery. It is a very, very dangerous stem cell therapy. Most of the countries they are using the stem cell therapy. India too. Hormonal therapy, chemotherapy, radiation. Our matter of concern is immunotherapy and targeted drug delivery system. This is the colorectal. This is the picture you can be seeing. The colorectal cancer is command a significant presence of the global cancer. Mortality rate is 100%, and the most common cause is older age. I really apologize. I'm not going to discriminate any of the people on the basis of their races, but in black races, the most common cause comparison to the white races. Again, because of their socioeconomic status, be there of late diagnosis, but there are so many different conditions. I apologize to be discussed later, but different kind of the things. About American and African area, the number of incidents comparison to the white races is 20% higher, and the mortality rate is 40% higher. Now, apart from that, the information involves the formation of cysts and polyps area. In the feature of concern, the colorectal cancer is developed nations. One can tell me why? The reason, because they consume the heavy junk food. They consume the food which is having the less fiber. They consume the food which is having the drastic damaging level of lipid. And even the report is also available that lipid is deposited at the rectal area. They deposit at the rectal area and the cells are growing over there. Once they cut and scissor the area, they simply lipid body collected from there. The doctors. So if we talk about the stages, it is having the 4 stages. The third stage is good to identify. And the first and second is very difficult to be identified. But when third stage, it is further divided into the 3 substages. Stage A, B, and C. The stage B and C is about to require the radiations or chemotherapy, but the stage 4 is completely incurable. Chemotherapy. Right? Our molecule, our defined molecule is on this particular. So we have used the nanotechnology to be overcome these challenges. सर थोड़ा सा पास है मैं चाहूंगा थोड़ा सा क्योंकि मैं और हम लेट थोड़ा सा other nanotechnologies you have heard about the different kind of the plant part, leaf extract, plant extract, root extract used for the nanotechnologies, but we are here not using those particular plant parts to be synthesizing the nanoparticle, there is something else, and that very you know. Have you heard anyone about the DXN? The DXN is company which is manufacturing the maximum products of mushrooms, and that mushrooms is having the huge medicinal properties as the ganodol. This is ganodol, a medicinally significant mushroom used by the Chinese, used by the Japanese, and so many worldwide, more than 150 companies using their products. Even one can we Google and see the products of DXN. The very nice products of the DXN available in the market, and they are having the property to immunoboosting, the weather protective, then anti-inflammatory, hypoglycemic activity, different kind of the activities it is having, and huge number of the different biomolecules contained. So what we did, we utilized the available knowledge of the Ganoderma, isolated its metabolite and polysaccharide content, and synthesized the nanoparticle, but the nanoparticles are not the regular one. They are the different nanoparticles, which was earlier using by the cancer treatment in a different way. This is just how about the review. We can go through the methodology part. The particle, the molecule that we have used for synthesizing the nanoparticle is bismuth. The bismuth is not at all utilizing as a treatment of cancer. It was the cobalt-16 isotope, which was utilizing as a radiation therapeutic tool for overcome the cancerous cells, myeloma cells, tumor cells. Accidentally, what the scientists had done, they had used the photo cell of this synthesized nanoparticle, which is known as bismuth oxide. Once they put it the parallel electrode kind of the photoelectric cells around the beam of cobalt 60, the percentage of efficiency of the radiation is higher 40% than the normal one. So the concept of bismuth oxide nanoparticles is generated from there. One of my students, Shikha Verma, work in the IGIB. She had worked on the bismuth oxide nanoparticle, but she didn't get cobalt. The ideas generated from there are fused together. So this bismuth oxide nanoparticle is synthesized by using the bismuth nitrate pentahydrate, dissolved in the deionized water, then subjected with the nitrate, and finally with the alkali, or in the place of alkali, we can also use the urea, the amount of urea. Then finally the cobalt nitrate used to fuse and isolate the conjugate bismuth oxide. But now I have used this nanoparticle, conjugate nanoparticle, which is chemically synthesized. Now directly this chemical is harmful. So an idea is generated in mind, why should this chemically synthesized molecule turns into the biochemical molecule, so that it has been easily acclimatized by the biological system of human or animal body. Right? So what I did, I used the polysaccharide and mycelia of the Ganoderma molecule and subjected with the artificially synthesized or chemically synthesized copper bismuth cobalt bismuth oxide conjugate. Now the physical characterizations were done on the basis of UV visible spectrophotometry, FTIR, for finding out how many different kind of the functional groups are there. X-ray reflections to find out the exact structures of the molecules. The scanning electron microscopy to find out the exact shape of the nanoparticles. The TM to find out the size of this nanoparticle. Anti-cancer activity has been determined by the cell culture maintenance, that is SCT 116, that obtained through the custody of National Center for Cell Science, NCCS Pune. One of my apprentices there, though they had started very new. So NCCS is performed to determine the life viability of the cell. The measurement of apoptosis inductions by using the two dyes, amphetamine and propidine. The cell migration assay has been performed. The colony formation assay has been performed. Measurement of the reactive oxygen species. Reactive oxygen species basically the oxygen which is responsible to cause the damage in the cells. So we had used the cell system to damage the cancerous cells. We had generated so many reactive oxygen species inside the cancerous cells. So the cancerous cells is ultimately died. See, cancerous cells neither be repaired. They never will adopt the normal function. It is a better way to scratch, remove, liquidate. The measurement of mitochondrial membrane potential, from where we get the energy by the food. The food in form of the ion, in form of the electrons, they get into the mitochondria. And mitochondria is ultimately the powerhouse of the body. So why not we damage the powerhouse so that the energy is not also gained by the cancerous cells? The way that we get attracted. The physical characterizations of the silver nanoparticles are function. The crude excess loaded nanoparticles is cleaner by UV spectroscopy. This is how the peak that we have obtained. The two ways. The one is mycelial X-ray, another is the crude polysaccharide by using the soft-rate condensation that we had used. The FDI is performed, find out the different kind of the functional group, that is OH band, carbon nitrogen switching, NH band primary. These are we found in the bismuth oxide nanoparticles. This is how the group drug TOH is found over here. The X-ray diffraction. The X-ray diffraction pattern obtained from the bismuth nanoparticle, both synthesized from the mycelia as the Black's diffractions, sir, if I'm not mistaken, from the mathematics side. So you can be better understand the grip of Black's diffraction. That is 2 theta. The detection position of the reflected radiation, X radiation is obtained and with the structures of this molecule. This is in case of mycelia, and this is one is in case of the crude transaction S77.33, if I'm not mistaken, 77.66. The scanning electron microscopy has been also performed with the conjugate cobalt and bismuth oxide nanoparticles. This is how the structure scanning, the superficial structure we got, the size of the shape of we got about the nanoparticles. And as far as this is for the nanoparticles crude body X-ray, the TEM gave the exact size. It is somehow looking as a slight triangular and it is looking how somehow slightly triangular and the quadrant size looks like positions over here. SED diffractions, when we bombard the electron beam and get reflected from the end, K, L, and N different orbital of the molecule. They emit the electron, so that electron is recognized by the detector and gives the beam. This is how about the bismuth oxide crude extract. Now, anti-cancer activity, we determine the entity assay. There is a tetrazoline salt, which gives the different color change when with the incubated with the cancer cells of the cell line culture. This is how the graphical presentation of the control positions. The 20 milli microgram per ml of sample incubated with the cell line cultures after 24 hours. You can see the crowd of cells, dizziness of the cells over there, and the white clearing cells is at 24 hours later. This is how the measurement of process. I would like to be discuss about the process. The cell is itself programmed to undergo die if it is not good to go, if it is not the normal cell, if it is the cancerous cell. But the cancerous cells do the mimic of normal cells. They behave like a normal cells. And that's why our immune system fail to recognize those cells. So apoptosis is a process which induces the cells to undergo to death. Once the cells is died, the system will clean. In case of the cancer, the cells is not directly undergo body apoptosis. The synthesized nanoparticles induce the process of apoptosis in the cancerous cell. So the cancerous cells undergo through the death. And once it is died, the debris of the cells is phagocytized by the normal macrophage cells or the phagocytic cells. If it is in the blood, say the leukemia. So if it is in the blood, it will be done by the monocyte. You can see the dye. This actinic dye colors the cancerous cells. And as the cancerous cells after the desired period of incubation is overdone, undergo to the death, the normal cells will appear, which is the colored by the red color to the propadium. This is with the another nanoparticles of 238. If we see the cell migration assay, we can see we have incubated the cell, all the cell line culture. One more thing that I would like to be clear is that SCT is the live cell line culture of carcinoma colorectal cancer, right? Cancer cells of colorectal region, right? And it is available in the market about 50 ml of live culture is 25,000 or something. So this is the effect of the mycelia, for the mycelia. This is again the conjugate cobalt bismuth oxide, and this one is for the wood scratch acid for the fruit extract. This is the colony formation acid. You can be clearly find out the number of huge colonies is over here. And after the incubation, 30 mg per ml or 25 mg per ml over the period of 24 hours, the number of carcinoma cells is very, very less. Similarly, the reactive oxygen is the same. So, would like to conclude that plant, which is somehow considering it as a plant, though it is not a plant, it's a major but more and more molecules, the more and more metabolites that's equal to the plant, which were investigated for the production of nanoparticles by using both its mycelium as well as the food body. The metabolite or the polysaccharide obtained from this mushroom is basically a capping agent. It is formed, the well understand that it is from a kind of a colloidal molecules around the nanoparticles. Basically, it chilled the nanoparticle inside. So once it is get into the body, either through the intraperitoneal or muscular or intravenous, the body system won't refuse it because it recognizes that polysaccharide is the substance of biological origin. And once this polysaccharide is digested, the nanoparticle will start to do its job. The cytotoxic experiment also showed the removal of harmful cells, and the sample were tested for anti-cancer activity, both for mycelial extract and polysaccharide. These are some major references I discussed. I think and especially to Dr. Shubha Suresh Kumar, Vice Chancellor, Savitha Institute of Technology and Medical Sciences, because I am from the microbiology and immunology background. So try to infer the carcinoma cells and how they are reacting against the antibiotics. So he helped me a lot to infer. And also to Solomon Basar, who had provided a lot of information about the polysaccharide reason. And I have personal relations during my PhD work. I have been contact since 2002. And thankful to P. Sarjan, who is basically from the Chennai and ambassadors in the King's College London. Though I don't have any direct touch with him, I just go through with Suresh Kumar. So he helped me a lot to why the cancerous cells act against any kind of the drug in a positive manner or they oppose the drug delivery systems Reason because they are having the different metabolic behavior as compared to the normal cells. They have a different homeostasis systems as compared to the normal cells. So this is just how about the small part. And we have 4-5 publications. The one patent we are going for that. We had already submitted the patent for this nanoparticles already. And we also have designed a system, a biological system to find out that if the nanoparticle is inhibiting the growth of cancer cells. So without any magnifications or fluorescent microscopy, is there any system, viable system, which can be simply confused at the 24 hours later or the desired period of incubation that this much number of the cells is decreases or died cancer cells and this much viable cells present in this system. So thank you very much, the organizing committee.
Thank you so much, Dr. Saraf sir, for a very nice and informative session. Is there any question from scholars and?
आपका क्वेश्चन मेरे लिए एनर्जी की तरह होगा क्योंकि काफी सारा अभी लेफ्ट हो गया मेरे।
Though I am not from the very beginning from presentation, so last year I have joined only few. So I only want to some references that you have stated that yes, the ganoderma has some type of specific chemicals which is actually related to or very much similar to plant. So if there is any references for this comment.
Yes. Say, there is scientific comparison. Of course, the related is the plants, so many medicinal plants having the phenols, terpenes, terpenoids. From the student side, anyone can tell me that have you heard about the terpenes and terpenoids?
So we know that is specific.
मैं आपसे एक क्वेश्चन कर रहा हूँ आप सब लोगों को पता होता है everyone knows the plant on the earth having the maximum amount of the terpenes and terpenoids we feel the very nice fragrance and the time has come गर्मी आ गई है आम आने वाला है तो आम का महक होता है ना वो जो चिपचिपापन होता है then because of the terpenes and terpenoids that greatly occurs in the plant and on the earth ganoderma is there. So many metabolites which is occurring in the plant are equally having in the ganoderma, though it is not having the photosynthetic property, though it is not having the actual metabolism behavior as the plant, but still it is containing the numerous number of the metabolites which is having in the plants. So that's why it is keeping in the category of the plant, not by the scientist, but by the researcher. They consider the ganoderma equivalent to the plant. No, it's not a.
We sincerely thank you, Dr. Ashish Saraf sir, for sharing your knowledge and perspective with us. Now, may I request our director, Dr. Jayant Maran sir, to felicitate our speaker, Dr. Ashish Saraf sir, with memento kit and certificate. Now, we move forward for the felicitation of today's esteemed session chair. I would like to invite Dr. T. Ramarao sir, Vice Chancellor, Anjaneya University, on the stage to felicitate Dr. Dhiren Singh sir. It is our privilege to honor and express our heartfelt gratitude for your valuable presence and thoughtful observations. I request you to kindly accept this small gesture of respect. Thank you so much, sir. I now hand over the mic to Minati Gora ma'am, Assistant Professor, for ending today's session. Thank you, ma'am. Respected dignitaries, VC sir, Director sir, Registrar ma'am, distinguished session chair, invited speakers, Dean of Science, and present HODs, faculties, and dear students. As we come to the end of technical session 1 of INCON 26, it is time to reflect on the insightful presentation and enriching discussion that have truly enhanced our academic experience. Now, I would like to request our respected head of the department, Dr. Sachin Kumar Das, to deliver the valedictory address for technical session 1.
Respected guests, respected speakers. First and foremost, I would like to express our deepest gratitude to our keynote speakers and panelists, your expertise, thoughtful analysis, and willingness to share your knowledge and enrich us. Our gratitude also goes to our sponsors and partners. Your generous supports made this conference possible and allowed us to create a